Imagine a future where cancer treatments are like tailored, totally personalized, hitting those cancer cells, but leaving the healthy ones alone. You know what I mean? It's not sci-fi either. There's this protein called SLFN11 that's like a key player in all of this. And yeah, that's what we're going to unpack today.
Yeah, it really could change how we think about cancer therapy, moving away from that one-size-fits-all to something way more precise.
Right. Doctors knowing exactly what'll work best based on your specific tumor. So to dive into this, we're looking at some really cool research from Dr. Maciej Kaczorowski and his team over at the Department of Clinical and Experimental Pathology at the Wrocław Medical University.
And their work. It just got published in the American Journal of Surgical Pathology. The title's a mouthful, but it sums it up well. Immunohistochemical evaluation of SLFN11 expression in cancer in the search of biomarker-informed treatment targets.
Okay, so SLFN11, maybe you've heard of it. It's basically this protein, super important for how cells react to DNA damage, like the kind you get from chemo and other cancer treatments.
Yeah, and get this, SLFN11 can actually stop DNA replication in those damaged cells. Basically, it makes the cancer cells self-destruct.
Whoa, so it's like they have this built-in, self-destruct button.
Exactly.
So if your tumor's got a lot of SLFN11, those cancer cells are more likely to just, go kaput when they get hit with these DNA-damaging therapies.
That's the idea. Dr. Kaczorowski's team looked at over 6,600 tumor samples from 127 different cancer types. It's a massive study.
Wow.
And they used immunohistochemistry, which is the best way to detect SLFN11 directly in the cells.
And this is where it gets really interesting, right? They found some patterns. Some cancers have tons of SLFN11, so they're probably going to respond super well to those DNA-damaging treatments.
Yep, like Ewing sarcoma and DSRCT.
Right.
Really aggressive tumors, but they almost always express SLFN11, so they're probably really vulnerable to treatments that, go after that pathway.
So for patients with these cancers, treatments that target this protein could be a total game changer.
Potentially, yeah. They also saw high SLFN11 expression in mesothelioma, clear cell renal cell carcinoma, and ovarian sericellus carcinoma.
Okay, that's encouraging. But what about cancers with low SLFN11 expression? Does that mean they're kind of out of luck?
Not at all. There's actually research going on right now to find ways to activate SLFN11 in those resistant tumors.
So they're trying to make those tumors vulnerable to treatments that wouldn't have worked before.
Exactly. It's pretty cool. One promising approach is using epigenetic drugs.
Oh, yeah. I've heard of those.
They can switch on SLFN11 expression, make the tumor sensitive to treatments that were useless before.
That's amazing. So SLFN11 research is really just getting started. What else is on the horizon?
Well, researchers are even trying to figure out how to bypass SLFN11 completely by attacking other molecular pathways involved in DNA damage response.
Wow. So even if a tumor doesn't have SLFN11, there might be other ways to sneak up on it and exploit its vulnerabilities.
You got it. It's like having a whole toolbox, each tool tailored for that specific tumor. This research is really a piece of the puzzle in figuring out how to treat cancer better. It really shows how important it is to understand what makes each tumor tick, at the molecular level. That way you can pick the best treatment.
So it's not just about discovering new drugs. It's about using the ones we have smarter.
Exactly. SLFN11 is giving us the map to do just that. But keep in mind, this is still early research.
Right.
We need more studies to make SLFN11 testing, standardized, and to really confirm that it works for different therapies. But Dr. Kaczorowski's findings, they're the foundation.
I'm curious. Could SLFN11 do more than just help us choose the right treatment? Could it tell us something about how different cancers work, their basic biology?
That's definitely a possibility. Researchers are looking into it.
So SLFN11 expression could give us clues about how different cancers develop and why they behave so differently. And that could lead to even better treatments down the road. Okay, so SLFN11 could totally change how we treat cancer, make it way more personalized. But I want to understand how Dr. Kaczorowski's team actually measured this SLFN11. They used immunohistochemistry, right? What's so special about that?
It's all about precision. Immunohistochemistry, or IHC, lets us look right at the SLFN11 protein inside the tumor cells.
So we're not just looking at the genes, we're seeing the actual protein in action.
Yep. Other methods might only look at the mRNA, which is the instructions for building the protein.
But just because you have the instructions doesn't mean you actually built the thing.
Exactly. And that's why IHC is so valuable. It tells you the protein is there and it's working.
So it's like making sure the house is actually built, not just looking at the blueprints.
Exactly. And that's why it's so reliable for making treatment decisions.
It's all about having the most accurate info to guide these personalized treatments, right?
Absolutely. This research shows how important it is to use the right tools when we're looking at these biomarkers like SLFN11.
So what about patients with cancers that don't have a lot of SLFN11? What are their options?
Well, like we talked about before, researchers are looking for ways to activate SLFN11 in those resistant tumors.
Right? Those epigenetic drugs that can like flip the switch and turn on SLF11.
Exactly. They're trying to make those cancers vulnerable again. And they're looking into other strategies too, like immunotherapy and targeted therapies.
Wow. So many possibilities. Like we're seeing cancer in a whole new way.
It is.
We're starting to understand its weaknesses and finding new ways to target them.
That's a great way to put it. This research is all about understanding what makes each cancer unique and tailoring the treatment to fit.
Okay? So even with low SLF11, there's still hope for effective treatment.
Definitely low expression just means we need to get a little more creative, find other ways to attack the cancer.
This brings us back to personalized medicine. It's not about finding one magic bullet. It's about having a whole arsenal and knowing which weapon to use.
Precisely. And biomarkers like SLFN11 are giving us the intelligence to make those decisions.
I'm really starting to get excited about this. But let's take a step back. What does all this mean for patients right now?
Well, the hope is that by understanding SLFN11 better, we can develop treatments that work better, help people live longer, and cause fewer side effects.
Sounds really promising. So where do we go from here? What's next for this research?
We still have a lot to learn. Scientists need to figure out how to standardize SLM11 testing, and we need large-scale clinical trials to make sure it really works.
So we need to validate these findings and make sure they actually help people.
Exactly. But Dr. Kaczorowski's research is a great starting point. This is where it gets really interesting. Beyond just choosing the right treatment, could SLFN11 help us understand the underlying biology of different cancers?
Oh, that's a great question. You mean like could SLFN11 expression tell us something about how different cancers develop and progress? And that knowledge could lead to even better treatments in the future.
Exactly. It's about understanding the why behind the what. Why do some cancers express SLFN11 and others don't? What makes them tick?
It's like cracking a code to find each cancer's weak spot.
Precisely. And once we know those weaknesses, we can design treatments that are even more precise and effective.
I can't wait to see what they discover next. But before we get too far ahead of ourselves, let's make sure we've covered the basics of SLFN11. Can you give our listeners a quick recap of what it is and why it's so important?
Of course, SLFN11 is a protein that plays a crucial role in how our cells respond to DNA damage, which is really important when we're talking about cancer treatment.
Because a lot of cancer treatments like chemo work by damaging the DNA of cancer cells.
Right.
Right.
And when a cell's DNA is damaged, SLFN11 can stop the cell from replicating its DNA, which basically stops the cell from multiplying and eventually leads to its death. It's like a checkpoint that makes sure damaged cells don't keep dividing.
So it's like a quality control system that gets rid of potentially dangerous cells.
Mm.
And when it comes to cancer, that's a good thing.
Exactly. And that's why SLFN11 is so important for cancer research. It could be a really powerful tool for personalizing cancer treatments so we can develop more effective therapies and get better outcomes for patients.
This is all starting to make sense. But before we dive into the specifics of Dr. Kaczorowski's research, let's take a quick break. We'll be back in a moment to explore the fascinating world of SLFN11 and how it might revolutionize cancer treatment.
So let's dig into what Dr. Kaczorowski and his team found about, SLFN11 expression in different cancers. They looked at over 6,600 tumor samples. It's a ton of data.
Yeah. So what really stood out to you about SLFN11? Anything surprising?
Well, one thing was the high SLFN11 expression in mesothelioma. Like, it was really consistent.
Mesothelioma, that's the one linked to asbestos.
Exactly. And they found over 90% of the mesothelioma samples had high SLFN11. So that suggests they could be really sensitive to those DNA-damaging treatments.
Wow. So that can be huge for mesothelioma patients, especially since it's often caught later on.
Absolutely. It just shows how important it is to test for SLFN11 early and accurately.
Yeah, for sure. What else did they find?
Oh, they also found high expression in clear cell renal cell carcinoma.
Okay, and remind me, what is that one again?
It's a common type of kidney cancer. And this finding suggests that DNA-damaging therapies, like chemo or PRP inhibitors, could be really effective for these patients, especially if their tumor has high SLFN11.
It's like having an extra piece of information to help make those treatment decisions, right?
Exactly. It's all about personalized medicine, tailoring the treatment to the individual.
Right. So what other cancers showed high SLF11 expression?
They also saw it in ovarian serous carcinoma, the most common type of ovarian cancer, which is unfortunately often diagnosed late. But this finding could be really significant for those patients.
It's like a new avenue for treatment.
Yeah, potentially.
Yeah.
Now, remember, they looked at both epithelial and mesenchymal tumors.
Right. Okay. So remind me, what's the difference between those again?
So epithelial tumors start in the lining of organs. And mesenchymal tumors, they come from connective tissues. And interestingly enough, they found that overall mesenchymal tumors had higher SLFN11 expression.
Huh. So does that mean they generally respond better to DNA-damaging treatments?
Well, not necessarily. It was higher overall, but there was still a lot of variation within the group.
So each cancer type is unique.
Exactly. You can't just lump them all together. But within those mesenchymal tumors, so like desmoplastic, small, round cell tumor, and Ewing sarcoma, they almost always expressed SLFN11.
Those are both pretty rare, right?
Yeah, and aggressive. And they mostly affect kids and young adults.
So this finding could be especially important for those patients.
Absolutely. Because it suggests those cancers might be really vulnerable to treatments that target this, SLFN11 pathway.
So this could lead to new and better treatments for these young patients.
That's the hope. This is how biomarker research like this can really change things for the better, moving towards more personalized care.
Absolutely. So we've talked about cancers with high SLFN11. What about the ones with low levels? What did the research say about those?
Well, they found low expression in some pretty common cancers, like hepatocellular carcinoma, prostate cancer, colorectal cancer, and breast cancer.
Those are some of the biggest ones globally.
Yeah, it is a significant finding. It suggests that these cancers might not respond as well to the traditional DNA-damaging therapies.
That doesn't mean there's no hope, right? Didn't we talk about some other approaches?
Exactly. There are other options, like those epigenetic drugs we talked about. They could potentially switch on SLFN11 expression.
So make those cancers sensitive to treatments that wouldn't work otherwise.
Exactly. And there's also immunotherapy, which uses the body's own immune system to fight cancer, and targeted therapies, which zero in on specific vulnerabilities in the cancer cells.
Oh, we're really starting to see cancer differently now.
Right.
We're understanding its weaknesses and finding new ways to target them.
That's a great way to put it. It's all about understanding the unique molecular profile of each cancer and using that information to choose the best treatment.
So even with low SLFN11 expression, there's still hope.
Definitely. It just means we need to be more strategic,
Yeah.
find other ways to attack the cancer.
Like a different battle plan.
Exactly.
This all comes back to personalized medicine. It's not about finding one magic bullet. It's about having a whole arsenal at our disposal and knowing which weapon to use for each individual patient.
Precisely. And SLFN11 is giving us the intel to make those informed decisions.
I'm also curious about the findings on the subtypes within each cancer type. Did they find any differences there?
Yes, they did. They found that even within the same cancer type, different subtypes could have different SLFN11 expression patterns, like in breast cancer, the triple-negative subtype, which is known to be very aggressive. It often had higher expression compared to the other subtypes.
Wow. So that could change how we treat triple-negative breast cancer.
It could. It's definitely a promising avenue to explore. Understanding these subtype-specific patterns could help us refine those treatment strategies and hopefully get better outcomes for those patients.
This is all so fascinating. It seems like SLFN11 isn't just a marker for treatment decisions. It's also giving us clues about how these different cancers work on a biological level.
You're absolutely right. The fact that its expression varies so much suggests that it might play a really important role in how those cancers develop and progress.
So we could potentially use it to figure out each cancer's weakness.
Exactly. And then develop even more targeted therapies. Attacking those weaknesses with pinpoint accuracy. Imagine what we could achieve if we could really unlock those secrets.
It's like we're on the cusp of a whole new era of cancer treatment, moving away from those broad-stroke approaches to something way more precise and personalized.
It's an exciting time to be in this field, and Dr. Kaczorowski's work is a huge step forward in our understanding of SLFN11 and its potential.
So SLFN11 is a game changer.
Yeah.
But there's still so much to learn. What are some of the big questions that scientists are trying to answer now?
Well, they're really digging deep into how SLFN11 interacts with other proteins involved in DNA damage repair. and how its expression is regulated in different cells. They're even looking at its role in cancer metastasis and drug resistance. It's a really complex and fascinating area of research.
It sounds like there's a whole universe of possibilities out there, and each new discovery could bring us closer to more effective cancer treatments.
Absolutely. This is really just the beginning. And as we continue to learn more about SLFN11, I'm confident that we'll see even more incredible discoveries that will change how we think about and treat cancer.
Before we move on, can you remind us how Dr. Kaczorowski's team measured SLFN11 expression? I know they used IHC, but why is that method so important?
Right. They used immunohistochemistry, or IHC, and it's so important because it's so precise. It lets researchers actually see the SLFN11 protein right there in the tumor cells.
So they're not just looking at the genetic code, they're actually visualizing the protein itself.
Exactly. Other methods might only detect the mRNA, which is like the blueprint for the protein, but that doesn't necessarily mean the protein is actually being made.
So IHC is like seeing the finished product instead of just the instructions.
Precisely. And it gives us that tangible proof that we need to make those informed treatment decisions.
Makes sense. It's all about having the most accurate information possible.
Yeah.
But what about those cancers that don't express a lot of SLFN11? What are the options for those patients?
Well, like we mentioned before, researchers are looking at ways to activate SLFN11 in those resistant tumors.
Yeah.
To make them sensitive to those treatments that wouldn't work otherwise.
Like those epigenetic drugs that can kind of turn on SLFN11 production.
Exactly. They're like finding a way to reprogram those cancer cells. And they're also looking at other strategies too, like targeting different pathways involved in the DNA damage response. It's like finding a backdoor into the cancer cell.
Wow, there are so many possibilities. It's like we're entering a new age of cancer treatment.
It really is. And SLFN11 is playing a key role in that transition.
But I imagine there are challenges too. What are some of the hurdles that scientists face when it comes to unlocking the full potential of SLFN11?
Well, one of the big ones is standardizing SLFN11 testing. Right now, different labs might use slightly different methods, which can lead to inconsistent results.
So it's like trying to compare apples and oranges.
Exactly. This lack of standardization makes it hard to compare data from different studies and to be confident about the results in a clinical setting.
So how do we fix that? What needs to happen to make sure everyone's on the same page?
It's going to take a team effort. Researchers, clinicians, and regulatory bodies need to work together to come up with clear guidelines for SLFN11 testing. Things like which antibodies to use, the staining protocols, and the scoring criteria.
Do we need a universal language for SLFN11 testing?
Exactly. That way we can be sure that the results are reliable and meaningful for guiding treatment decisions.
This is all so groundbreaking. But before we get too carried away, I want to make sure we understand the clinical significance of those different SLFN11 expression levels. We know that higher expression is usually better, but how do we know what levels actually predict a good response?
Right, that's another challenge. We need to figure out those precise cutoff points. More research is needed to establish clear thresholds that can guide treatment decisions. And that research will probably involve looking at SLFN11 expression alongside other clinical and molecular factors.
So it's about putting all the pieces of the puzzle together.
Precisely. And the more we learn, the better we'll be able to tailor those treatment strategies to each individual patient.
It's really exciting to see this shift towards more personalized cancer treatment, moving away from that one-size-fits-all model.
It's a true paradigm shift, and SLFN11 is right at the heart of it.
This has been such an incredible deep dive into the world of SLFN11. I've learned so much, and I'm really hopeful about the future of cancer care.
Me too. It's amazing how far we've come in this field. And I have no doubt that there are even more amazing discoveries just around the corner.
Welcome back to the Deep Dive. It's been quite a journey exploring SLFN11 and its potential to, change how we treat cancer. And as we wrap things up, I want to talk about the challenges ahead and what the future might hold. So what are some of the big questions that researchers are still wrestling with?
Well, one of the biggest challenges is, like you said earlier, figuring out exactly what different levels of SLF expression actually mean. Clinically, we know that more SLFN11 is generally better, but how much is enough? What's the magic number that tells us a patient will respond well to a specific treatment? We still need to pinpoint those thresholds.
So it's not just about whether it's there or not. It's about the amount.
Yes. And it's likely that SLFN11 expression will need to be considered along with other factors, things like the patient's overall health, the specific type of cancer, and other molecular markers. It's like putting together a puzzle to get the full picture.
And speaking of the full picture, can you remind us again why immunohistochemistry, or IHC, is so important for measuring SLFN11 accurately?
IHC lets us see the actual SLFN11 protein inside the tumor cells. It's like looking at the finished product instead of just the blueprints.
Right, so not just looking at the genetic code, we're seeing the protein itself in action.
Exactly. Some other methods might only detect the mRNA, which is like the instructions for making the protein,
Yeah.
but that doesn't tell us if the protein is actually being produced.
It's like having the recipe but not knowing if you actually baked the cake.
Exactly. IHC gives us that confirmation. It's the gold standard because it shows us the protein right there in the tumor cells, so we know we're getting accurate information.
So IHC helps eliminate doubt and make sure we're targeting the right cells for treatment.
Precisely. It's all about having the most reliable information to make those treatment decisions.
This really underscores the importance of using precise and reliable tests when it comes to personalized medicine.
Absolutely. And Dr. Kaczorowski's research really highlights the value of IHC for getting an accurate read on SLFN11 expression. It's a crucial tool for moving towards that personalized cancer care that we've been talking about.
Okay, so what's next for SLFN11 research? Where are scientists focusing their efforts now?
There are a lot of exciting avenues to explore. One big one is standardizing SLFN11 testing so that different labs get consistent results.
Right, because right now there's some variation in how different labs test for SLFN11.
Exactly. It's like using different rulers to measure the same thing. The results won't be comparable.
So we need a universal standard for SLFN11 testing.
That's right. So that we can be confident that the results are reliable and meaningful for guiding treatment decisions.
And what about those cancers that don't have much SLFN11 to start with? What's on the horizon for them?
Researchers are working hard to find ways to activate SLFN11 in those resistant tumors. One promising approach is using those epigenetic drugs that we talked about earlier. You know, the ones that can switch on gene expression.
So it's like finding a way to turn on the SLFN11